Water scarcity is an increasing concern around the globe. In fact, by 2025, it's estimated that over half of the world's population will be living in a water-stressed area. In order to meet our increasing water needs, we must be able to clean our polluted water. A class of water pollutants that are becoming increasingly problematic are something called cyanotoxins. Cyanotoxins are these dangerous chemicals that are produced by harmful algal blooms. But although we call them algal blooms, they're more often caused by a similar looking organism known as cyanobacteria, which is why we call the toxins cyanotoxins. In particular, I study one of the most common cyanotoxins known as microcystins. Microcystin is a potent liver toxin responsible for a variety of livestock and pet poisonings, as well as human illnesses every year. Short-term exposure to microcystins can result in things like rashes, nausea, and vomiting, but long-term exposure can result in liver failure and may even cause cancer. This becomes really problematic when source waters containing a bloom are taken into drinking water treatment plants. In this case, both the cyanobacteria and cyanotoxins are taken into the plant, and if not treated effectively, may end up in tap water making people sick. Unfortunately, our current treatment strategies aren't always sufficient to stop this from happening. But this is where my research comes in. I'm investigating the use of a technology known as electron beam irradiation technology, or ebeam for short. But don't let this irradiation term scare you. The way that this technology works is essentially by taking electrons from electricity and speeding them up almost to the speed of light. This laser-like beam of electrons is then able to interact with the chemical bonds on pollutants to break them apart, as well as to damage the DNA of harmful bacteria and viruses. My research questions whether we can use this technology to both break down microcystins in drinking water, but also to kill the cyanobacteria responsible for producing them. So far, my research has shown that after irradiation, microcystin is broken down into non-toxic products. Normally, microcystins interact with enzymes in your liver cells, known as phosphatases. But after irradiation, we see that the part of the molecule responsible for this binding is no longer present. This suggests that we've gotten rid of the toxicity of microcystin. But why do we care if the cyanobacteria are still there producing more microcystin and putting it into our water supply? Well, I also see that after irradiation, cyanobacteria cells lyse, or are essentially killed through the rupturing of the cell. This means that they're not able to produce more microcystin. So overall, ebeam could be a valuable tool implemented into the water treatment process to stop pollutants like microcystins from reaching your tap.